Alrighty, hello everybody, and welcome to the video. Today we're going to be talking about a very exciting topic, the mechanism of muscle contraction. So what happens when you do a biceps curl? What's going on? It's very small, I know. What's going on in that muscle, with the muscle fibers themselves for a contraction? So it's a very detailed process, and we're just going to be talking about the general overview today. You're not expected to know every little minute part, and we're not going into great detail when it comes to things like the action potential and firing of voltage and so forth. All right, so today we're going to look at the process of skeletal muscle contraction. So what's going on in the events leading up to contraction? What I want you to take away from this is, can you write out the steps? You know, this happens and this happens and this happens and so forth. Then we're going to look at the importance of ATP in a muscle contraction. ATP is required for a certain step in the muscle contraction. Without it, you get what's known as rigor mortis, where the muscles are stiff and locked because they can't release and relax. So then we'll go over how a muscle relaxes and as well. So alrighty. So this image here goes through the general overview of a contraction. So here we have a muscle fiber, and here we have a neuron. Every single muscle fiber is served by its own motor neuron. So neuron is part of the nervous system, and it sends a signal. That signal is called an action potential, or AP. The action potential runs down this neuron. I know we haven't talked about the nervous system yet and how action potentials work, but just note that it's an electrical signal and it's sending a message. So that comes down here and releases that message at the end of that terminal. It's called an axon terminal here, and for a muscle, that's known as the neuromuscular junction. So right here, neuromuscular junction. So the action potential gets there, and now what happens there? So here, this box is zooming in to what's happening. So let's slide back over here. So here, in that neuromuscular junction, you have these little vesicles that are packaged with a chemical. This chemical is known as ACH, or acetylcholine. Acetylcholine is a neurotransmitter. A neurotransmitter is a chemical transmitter of the nervous system. So it sends a neurotransmission. So it's sending a signal. In this case, acetylcholine is excitatory, meaning when it's released here, so when the action potential travels to the end of this neuron, you get a rush of calcium coming in, and this is part of the steps where you don't have to worry about the intricacies of what happens. And that calcium then causes these vesicles to release neurotransmitter. Acetylcholine then comes into this gap. This gap is known as the neuromuscular junction. So it's a space. So the acetylcholine has to travel to that space, and then it communicates with the sarcolemma, or the cell membrane, of that muscle cell. So this would be the cell membrane right here, and it makes all these little folds called junctional folds to increase the surface area on where it can have these receptors. So these are acetylcholine receptors. So they bind acetylcholine and they open up the channels. They allow sodium to come in. So sodium comes into the muscle cell, leading to something known as depolarization. In brief, depolarization ends up exciting that muscle and leads to another action potential. So another electrical signal traveling down that sarcolemma of the muscle, and eventually leading to, excuse me, a contraction in that particular muscle. This neuromuscular junction here, where all these acetylcholine receptors are, is also known as the motor end plate. And this event here where sodium comes in is known as end plate depolarization. So end plate depolarization leads to an action potential, and once you hit that action potential, that muscle will contract. So that excitation wave travels down through the muscles. It goes down those T tubules, which, remember, you don't want the wave to just go around the outside of a muscle cell, because then you're only going to release calcium from the outside, and you're never going to contract the interior muscle myofibrils. So the T tubules come down into the middle and then allow calcium to eventually be released from the middle of the muscle cell. So that's what this is showing here, this T tubule coming down in, and then alongside that T tubule, you have the sarcoplasmic reticulum, or the SR. Remember, the sarcoplasmic reticulum is a modified smooth endoplasmic reticulum, and here we have calcium. So calcium 2+ is stored in there. And so as this action potential works its way down, calcium gets released from that SR. So calcium then floods out through the intracellular space. Excuse me one second. And then why do we have that calcium out there? Well, that calcium is what combines this excitation event to this contraction event. It's called EC coupling, or excitation-contraction coupling. So how do we combine this excitation event right here to contraction? Well, it's known as EC coupling, or excitation-contraction coupling. So what does this calcium do now? So if you recall, the myofibrils are made up of these two filaments, thick and thin filaments, actin and myosin. If we go down here, this is a refresher of those different filament lines we have. So remember, actin is the thin filament right here. It's just the two strands wrapped around each other. And then the thick filaments are the myosin right here. Remember, you have the I bands, which are just actin. You have the darker A bands, which is where you find the myosin here. And then you have the Z discs, which are filaments that are holding the actin together and form these little Z lines. You have the H zone that's just myosin, and then there's an M line, which is the middle of that, where there's a protein that helps hold the myosin together. There are other proteins involved here, but this is just the gist of it. So when a contraction happens, these myosin heads need to bind to the actin. There are binding sites on actin, those little black dots right there, but you can see they're blocked at this point. But myosin binds to that and goes through something called a power stroke, where it pretty much just goes like this and moves like that. So you can imagine that pulling the actin in. So myosin slides over the actin, and that leads to a shortening. So here you see the binding sites are open. Myosin is bound to that, and it goes through a power stroke, and then you see a shortening. So here was a Z line, Z disc, but now you see it over here. So there's a shortening. That H zone disappears now, because now you have actin in there, and everything shortens, and that's what a muscle contraction is. But how do we control this power stroke event? And that's where calcium comes into play. So here, calcium comes in, and there's these two proteins. They're two T words. There's the globular unit, which is called troponin. And then there's the fiber, this orange fiber right here, that's called tropomyosin. So tropomyosin is usually blocking those myosin head binding sites. But what happens is calcium binds to troponin. You see the two little white dots signifying calcium comes in and binds to that. And what that does is it causes a shape change and moves tropomyosin away from these binding sites, and then allows the myosin heads to bind and then go through the power stroke. And then everything shortens and you get a muscle contraction as a result. But there's still some more details we need to fill in there for the importance of ATP. I have no water, but I'll get through this. So here, now we're going to talk about ATP and the power stroke. So ATP is required for muscle contraction. Why? At what stage is it needed? So here we have calcium. We have the binding sites open. Myosin heads are bound. Now you see this ADP and a phosphate. What happens is these two leave. When they leave, that causes the myosin head to go from this to this. That is known as the power stroke. You see when it does that shape change, it causes it to shorten. It pulls the actin in. So you see the actin being pulled this way when these ADPs and this inorganic phosphate are released. So it gets pulled in and leads to the power stroke. Now we have to reset this. It's stuck in that in that position, not cocked position, but power stroke position. In order to release it, we require ATP. And so here, this is where ATP comes in and attaches to the myosin head. When it attaches to that myosin head, it releases it. It then goes through hydrolysis where it splits off a phosphate. So it becomes ADP, adenosine diphosphate, and an inorganic phosphate, and it resets it into the cock position and it's ready to go. If those binding sites are still open on actin, it will rebind and go through another power stroke, releasing the ADP and the inorganic phosphate. And then if ATP comes in, releases again, and it continues through that cycle. And that's what cross-bridge cycling does. So here, boom, it binds again. It recocks the myosin head into the cock position. So right now at rest, all your myosin heads are in the cock position ready to bind. The only thing preventing a muscle contraction from happening is the control of these binding sites right here. That's why calcium is required. When calcium comes in, it binds to troponin, moves tropomyosin out of the way, and those binding sites then open. Now, how do we stop this? How do we relax? Well, for relaxation, you stop the action potential coming down from that neuron. So your brain says, no more muscle contraction there, relax that particular muscle. So when that happens, you stop releasing neurotransmitter. When you stop releasing the acetylcholine can also diffuse away. There's an enzyme called ACHE, acetylcholine esterase, which can break down acetylcholine as well. But you don't get an action potential then being sent down that muscle cell. No action potential being sent down that muscle cell means no more calcium is being released. So calcium ends up being reuptaken into the SR, that sarcoplasmic reticulum. Calcium gets reuptaken. No more calcium is bound to troponin, and that tropomyosin then blocks the binding sites again. If those binding sites are blocked, the myosin heads can't bind, and you get an elongation and relaxation of that muscle. Now, all of this also takes energy. So after you're done using a muscle, there's still additional oxygen required to get the calcium stores back and reset that muscle. But then the muscle lengthens and relaxes. So there's a lot more to this. Just want you to understand that. This is the brief and dirty overview. Such as other stuff we could talk about is, where does that ATP come from? So like the first 15 seconds of a workout, you actually get that ATP being remade from something called creatine phosphate. And then there's a difference between anaerobic and aerobic respiration. So you can go for a run at a low heart rate or a walk for a very long period of time because you're doing what's called aerobic respiration, meaning you're making ATP in the presence of oxygen, and you make more ATP then. And then if you start sprinting, though, you go anaerobic, where you run out of oxygen and you're only making 2 ATP via glycolysis. So your muscle has to stop working faster, and you end up fatiguing sooner as a result of that, because of that lack of oxygen. And there's way more to it than that we're not going to get into here. But this is just a general overview of a muscle contraction. And I hope it was brief enough and easier to understand, since I didn't go into the final details of it all. But it's a really, really neat process of how your muscles contract. It's not as simple as just, oh, you pick something up. There's a very fine-tuned response for how it all works. But anyways, that's all I have to talk about today. Hope you enjoyed this video. If you have any questions, like always, let me know. All right, have a great day, everyone. Bye-bye.